High-throughput screening, or HTS, is a powerful methodology used extensively in drug discovery and biological research. It enables the rapid testing of thousands, even millions, of chemical, biological, or genetic compounds. The primary goal is to identify substances that exhibit a desired biological activity against a specific target. This automated process significantly accelerates the identification of potential drug candidates or probes for biological pathways. HTS has revolutionized how we search for new medicines and understand complex biological systems. It moves beyond traditional one-by-one testing to a parallel, miniaturized approach. The HTS process typically begins with a well-defined biological assay, which is a test designed to measure a specific biological or biochemical event. This assay is then miniaturized and adapted for automation, often performed in multi-well plates, such as 96, 384, or 1536 well formats. Robotics and liquid handling systems precisely dispense the assay components and the compounds to be tested into these plates. Each well contains a small reaction volume, allowing for numerous parallel experiments. After an incubation period, a detection system measures the assay's outcome, such as fluorescence, luminescence, or absorbance. Data is then collected, analyzed, and processed by specialized software to identify hits, which are compounds showing the desired activity. The success of HTS heavily relies on the availability and diversity of compound libraries. A compound library is a collection of chemical or biological substances stored and organized for screening purposes. These libraries are meticulously curated and often represent a vast chemical space. The quality and diversity of these libraries directly impact the likelihood of finding novel active compounds. They can range from collections of small molecules to complex biological entities. Each compound in the library is typically cataloged with its structure, purity, and other relevant data. One common type is the small molecule library, comprising synthetic organic compounds. These are often designed to be drug-like, meaning they possess properties suitable for oral bioavailability and target engagement. They can include diverse sets of compounds or focused collections targeting specific protein families. Natural product libraries are another crucial category, derived from plants, microbes, or marine organisms. These compounds often exhibit unique and complex chemical structures, having evolved to interact with biological systems. They have historically been a rich source of therapeutic agents. Beyond small molecules, peptide and protein libraries are also employed. These can be synthetic peptides or recombinant proteins, used to identify modulators of protein-protein interactions or enzyme activities. They offer different binding characteristics compared to small molecules. Furthermore, nucleic acid libraries, such as siRNA or CRISPR-guide RNA libraries, are used for genetic screening. These libraries enable researchers to systematically knock down or edit genes to understand their function or identify genetic vulnerabilities. This approach is powerful for target identification and validation. The compounds within these diverse libraries originate from several sources. A significant portion comes from synthetic chemistry, where chemists design and synthesize novel molecules in the laboratory. This allows for precise control over chemical structure and properties. Natural products are isolated and purified from biological sources like plants, fungi, bacteria, and marine invertebrates. These organisms produce a vast array of secondary metabolites with diverse biological activities. Advanced extraction and purification techniques are essential here. Combinatorial chemistry is a powerful method for generating large numbers of diverse compounds rapidly. This involves synthesizing collections of compounds by combining different building blocks in various arrangements. It efficiently explores chemical space. Finally, computational design plays an increasing role. In silico methods are used to design molecules with predicted properties or to identify potential hits from virtual libraries. These computationally designed compounds are then synthesized and added to physical libraries for experimental validation.